speaker this evening. Our speakers are going to talk for, roughly speaking, 25 minutes on the subject of their synthetic biology project. This evening's speakers are part of Professor Fiona Meldrum's team. So presenting, we've got Paul Turner and Daniel Burt. And they'll be talking, as I said, about the synthetic biology project. And then, when they've done that, there will be a break during which the venue would like me to encourage you to refill your glass. And then after the break, you'll have the opportunity to put questions to the team speakers. And just before we start that, 2 or 3 other things to mention. First of all, if anybody would like to write a review for the website, then please do so. Now, a review doesn't have to be an account of what's happened. It could be your opinion of one particular thing that was said or not said, something that you agreed with or disagreed with, or just it could be covering one aspect of what's spoken about this evening. On Cafe Scientifique websites of cafes scientifiques around the world, Brazil, Portugal, Australia, literally every country, you can read their reviews of who they get to come along to present and what they think of the topic, that sort of thing. So if anybody would like to do that, then feel free. And then maybe next time, October, November and so on, one or two people could come along prepared to write a review. If anyone would like to take notes and write a review, then feel free to grab a pen and a notebook. Okay, if you've already got a pen and a notebook, that's fine. Okay, I also need to say that I'm at the interval. I'll be handing the hat round for donations. We have expenses that we need to cover, including, of course, the cost of this venue. Right. Finally, before they start, if you received an email inviting you along for this evening, that's great. If you would like to receive an email inviting you along to future meetings, then you can write your email address down on this notebook, and then you'll get invited to future meetings. Or you can keep up to date with future meetings on the website, which is Leeds Cafe Sci. Put that into Google and you'll find it. Or you can also follow Leeds Cafe Sci on Twitter. Right, that's enough from me. So I'm going to hand over to Paul and Daniel immediately.
Good evening, everybody. My name is Daniel. Today I'm going to talk about synthetic biology. This is a really interesting time for synthetic biology. The government has just pledged £60 million worth of research funds for it, and it's estimated to be worth £7.2 billion by 2016. I'll begin this presentation with an overview of synthetic biology and the basic principles and technologies involved. After which I'll move on to the iGEM competition which we're competing in and tell you a little bit about that. Like most emerging fields of science, there are some concerns for the public. My colleague Paul will address some of these ethical and safety issues at the end. So what exactly is synthetic biology? There are many variants of a good definition, but I think this one's a good one. So, the design and construction of biological devices and systems for useful purposes. So it's very much an engineering field. We're trying to create functional biological devices. And it encompasses many current scientific fields, including but not exclusively, biochemistry, molecular biology, genetic engineering, cellular biology, and computing. So there's a lot of similarities between synthetic biology and computer programming. So when you have a problem, you create some software and it solves the problem. Similar fact, you can modify bacteria to solve problems. It's very exciting, very interesting. Genetic engineering is fundamentally taking genes from one organism, or the things that code for proteins, and inserting them into another organism to produce that protein, or show that characteristic. So this is an overview of genetic engineering, which is really fundamental in synthetic biology. So, again, a gene is a section of DNA which is the universal code for life. And each gene tells the organism how to make a specific protein. and proteins control everything in living systems. The principle of genetic engineering is to modify this code of life and make it have a specific function. Let's look at the production of insulin, for example, which is a big issue for diabetics. So you take the gene from the organism you want. In this case, they took human insulin. So you take it from a human donor, and you place it into a bacterial host. This is step 2 over here. Now, once you've got it in the bacterial host, I'm sure you guys have left milk in your fridge too long. You see how fast bacteria grow up. So you can easily and quickly grow up a whole colony of bacteria, so loads of them, and they'll start producing your insulin. And then all you have to do is purify out the insulin or bacteria, depending on what you want, and you have a product. It's like a mini microfactory. And we refer to these bacteria as genetically modified organisms. You might have heard the term GMO before. And this kind of research started in the late 70s and really pioneered genetic engineering, and is the foundation of what we research. So I'd like to talk about some of the technologies involved in synthetic biology, how we actually do our job. So, first thing, DNA sequencing. Basically, it's working out the genetic code, it's working out the sequence. And this is really cool because in the future you could have tailored medicines for your particular genetic code or your makeup. So tailored medicines just for you. Synthesis is actually taking the genetic components and putting them together, artificially creating our own genomes or our set of instructions. Modelling, using computer software to predict what's going to happen, a very important part. Without computer software, it would be near impossible to do half of these genetic engineering and synthetic biology processes. And most importantly, of any scientific research, measurement, actually getting data. And 3 and 4, modelling and measurement, are very important together. Because if you create a computer model and trying to predict how something's going to work, you want to check it with actually what happens and work out the discrepancies. So I'm going to talk about 3 examples, 3 problems in the world, and how synthetic biology has taken an approach to solve them. So my first one is vitamin A deficiency. The vitamin A deficiency is estimated to kill 670,000 children under the age of 5 a year, and most of these are from developing countries. How can synthetic biology provide a solution to this? Potentially a bit of agricultural alchemy, talking about golden rice. So rice naturally contains a chemical called beta-carotene within its leaves. Beta-carotene is one of the ingredients the human body uses to produce vitamin A. However, I hear rice leaves aren't too tasty. So scientists at the Institute of Technology, the Swiss Institute of Technology, have genetically modified the rice plant because the beta-carotene is produced in the endosperm, which is the edible part of the rice, giving it its golden colour and hence its name. So, although golden rice was developed as a humanitarian tool, it has been met with significant opposition from environmental and anti-globalization activists. My colleague Paul will talk more about that later. They're kind of expanding this research by adding more vitamins and more minerals into the rice. So in theory, you could create super nutritious foods, which could help with a lot of deficiencies and problems, dietary problems. The next problem I would like to talk about is biofilms. What are biofilms? Biofilms refer to communities of bacteria that clump and adhere together, so they glue themselves together. Why do they do this? They do it to protect themselves in really extreme environments. For example, if there's antibiotics present or extreme temperatures. If there's antibiotics, by clumping together, only the surface bacteria get affected, so the inner ones are protected. It's a survival mechanism they've developed. But it causes all sorts of problems, especially in industry and health. A lot of diseases are caused by biofilms, and they're really hard to get rid of with antibiotics, and take a lot of rough courses of antibiotics to get rid of. They do this by a phenomenon which is really interesting called quorum sensing. It's just a fancy term for chemical language. So bacteria can actually talk to each other using these chemical molecules. So one bacteria decided it wants to set up a biofilm, it detects all the antibiotics, and it will release a chemical. And the other bacteria will absorb this chemical and respond to it, just like you respond to the language when someone tells you to do something. Well, you probably won't do it, but if they don't, you're good. So, how can synthetic biology solve this problem of these viruses? How can it stop these bacteria clumping together and making it a real pain to destroy them, pretty much? So, the solution. So, researchers created a synthetic virus, so a genetically modified virus called a bacteriophage. A bacteriophage is the fancy name for viruses that infect bacteria. They modified this bacteriophage to have a gene which codes for an enzyme to break down the biofilm. So this enzyme, when it's produced, will cause all the bacteria to fall apart. They won't clump together anymore. But the beauty about a virus is it also codes for the virus itself. So as you can see here, the virus infects the first bacteria, for example this one here. And it causes the bacteria to produce loads of the enzyme to break down the biofilm, but it also produces loads of the virus. So eventually this bacteria will explode and release all of this enzyme to break down the biofilm, but loads more virus to infect all the other bacteria. So it's what we call an amplification mechanism. It amplifies itself. So the more it infects, the more viruses it produces, the faster it can break down biofilms. And it had a 99.7% removal rate of biofilm mass. So this is a great success. It's a really good example of synthetic biology actually working in the field. So the next problem I want to talk about is a little less devious, a little less severe. But basically researchers are looking into how can you control bacterial patterns on the micro scale. How can you control where they go with accuracy? So researchers created an ultra high definition, two-dimensional chemical image. So they created an image basically using modified E. coli. E. coli is just a very common strain of bacteria. So what relevance does this have to anything? So as you can see on this picture here, it's just kind of a schematic. So they shined light down onto this bacteria colony they had. And wherever the light was masked, so it's dark, it caused the bacteria to produce an enzyme which makes a dark pigment. So wherever it's dark, in the image, it transfers the darkness. And it'll be dark at that point. Wherever it's light, there are no enzyme produced, there's no pigment, so it's white, it's lighter. So you can build up a contrast using that. So this essentially is kind of like a biofilm, but in more sense of a movie film sort of application. So this technology is really cool if you wanted to create images, but it implies something I think that's way more interesting and way more potentially viable technology, which is you can use them to create you can use them to form microstructures. So you can create bacterial structures on the micro scale. Why is this useful? Because you can create computer chips, for example, or you can create tiny little factories that produce items, for example. So it's really, really cutting-edge technology here. This is one of the images they actually produced, trying to suck up to the journal they published in, I think. And yeah, that's it. And so now I want to talk about the competition we're involved in, which is called iGEM. It stands for International Genetically Engineered Machines. So, iGEM was originally started by MIT, Massachusetts Institute of Technology. However, last year it became a non-profit independent organisation. The aim of the iGEM competition is to develop and improve the field of synthetic biology. So the main way it does this, and the main idea behind it, is to develop a massive database of genetic parts. So people like every university team around the world will submit a part. They'll do research, they'll work something out. For example, one of the parts, or biobricks as they like to call them, could be the insulin part. So if you wanted your bacteria to make insulin, for example, you could go to this database, take the part for insulin, and then insert it into your bacteria. So it's kind of very modular, so you can chop and change things very easily. And it's very standardized, so everything fits together worldwide. Everyone can do this and actually efficiently achieve it. So just like the Industrial Revolution, before they used to have different types of screws with the same application, and then they standardized the screws. So every screw for that application had to be the same type, same size, and this promoted growth. And it makes sense, obviously, because if everyone's on the same page, it's a lot easier to get things developed. So there's 4 main divisions of the competition. High school, which is it's obviously not too intense because they're still in high school, but it really inspires younger students to get involved in synthetic biology. Collegiate, undergraduate, which is what we're doing right now, with over 100 universities from around the world competing in this. And every year we go to a jamboree, it's hosted in a different university, and we kind of present our research and collaborate on results. The laboratory, which is the professional labs, which I think shows a good community effort. They look past profit, and they're trying to work together to develop the field as well. And it's non-profit, obviously, so they're all submitting these things out of charity. And the 4th part is software, which I mentioned before is really important. So developing the tools to design all these genetic components and work out how they're going to work. And next year, they're introducing a DIY section, a do-it-yourself section. So people, enthusiasts, non-scientific professionals are actually in their garages and basements buying equipment and doing these projects themselves. Kind of like computers used to be in the 70s, where you get your little old computer and you create a little hello world application. It's very much like that at the moment. So let's talk about our project. Our project name is the Micro Beagle. And what we're trying to do is develop foundation research for a biosensor, so a biological device that senses something. And ours is looking at water technology, so we're looking to detect diseases within water. So why microbeagles? It's a bit of a weird name. So a beagle would hunt a hare, so our microbeagle hunts pathogens, which are disease-causing organisms. What makes our biosensor so special? This is basically how it works. So we, instead of using a pathogen or a disease-causing organism, we use silica, because obviously we're undergraduate researchers and it's quite dangerous to work with dangerous pathogens. But more than that, you kind of, when you start research, you want to start with proof of concept. You want to show that it works. And you want a system where you can take things out and replace them. So in theory, you can take this out and replace it with the pathogen. It's a modular system. What happens is, this is our microbeagle here, this big blue thing. And all the red things are genetic components that we put in. And what happens is the silica will bind to a receptor on our microbeagle, something that will detect it. And it will cause a chain reaction within the cell, lots of chemical reactions, which produce a fluorescent protein called GFP, green fluorescent protein. And basically, in mass amounts, that will make the sample glow. So if your disease is there, the water sample will glow. So it gives a very easy response. So what are the advantages of our biosensor? So we looked more at like developing world applications. No need for electricity. Perfect. Finding electricity in a lot of these developing world is hard. So no electricity, that means it's cheaper to run, easier to run. No need for complex analysis. If it glows, there's dirty water. If it doesn't glow, it's not dirty water. It's very easy to understand and interpret. You can use different colours to create a traffic light system. So they discovered a whole set of these fluorescent proteins. You've got yellow fluorescent, red fluorescent. So you can have different diseases fluorescing different colours. So it can give you a lot of information about a water sample just from one biosensor device. And it's cheap to produce. It's a bacterial culture. You can just take some off, grow it up, and pass it around. It's very nice. One major part of the competition we're doing is outreach and collaboration. So it's not just about doing science research all the time. It's about engaging with the public, engaging with other teams, and really trying to develop the field as a whole. So we're currently working with Purdue and Manchester. With Purdue, we're trying to standardise the iGEM database a lot better. And with Manchester, we're just doing some computer modelling. It's very geeky, very boring. Outreach and collaboration. So we've been engaging schools, and we've been trying to really inspire and educate children about this field, because hopefully it will be a big part of the next generation's future. So it's important to properly educate and inform the next generation. And we've also developed a lot of tutorial videos. We have our own YouTube page for both researchers and non-scientific professionals as well. So one last thing I'd like to talk about, is a little different, but it's quite interesting, is art. There's a massive movement within synthetic biology regarding art. So this was from the first synthetic biology and genetic engineering ARC exhibition. And you can see on the left, this guy here is smelling the questionable liquid. It's actually a bacteria that's genetically engineered to produce banana oil. So it's bacteria that smells like banana, which is pretty cool. And on the right here, I was talking about biofilms earlier. So this is a living biofilm, obviously, but they've modified it so it reflects light off the top. So it's a living mirror. And that's pretty cool technology as well. And thank you for listening to me. I'm going to pass on to my colleague Paul, who's going to talk about the safety and ethical issues involved with this technology. Thank you.
As Dan said, I'm going to talk about health and safety and things, as well as ethics. When it comes to synthetic biology, you can't really separate the two. They sort of go hand in hand, and I'm sure everyone's familiar having to fill in safety forms and the like, and many boxes. And we do that in science as well. It's important to do it in science. We try and avoid doing dangerous things, and when there is something that's dangerous, we try and do something that might reduce that risk. But we often talk about things called effective measures. So what we mean by that is something that either completely removes that risk, or is going to be a lot better than just putting on a pair of gloves. Like ideally have a robot do it would be good in science. It's what we do in space all the time. It's rather dangerous. So we do the same sort of thing here. So I thought I'd take you through what we've done as a team when going through these sorts of forms. So I'll start with the first one. So the first issue I'm going to bring up in terms of safety is containment. So lovely fields like this. I mean, what if we allow something we've modified to get out into the wild? And golden rice is a wonderful example of that. Golden rice is something that are literally asking people to replace their normal cultivated rice strains with a modified organism in the wild. It could easily spread anywhere, it could easily pass into other systems, and it could potentially damage them. At the same time, with it being a cultivated crop, you could argue it's already a modified ecosystem that's going into, but that's a whole other kettle of fish. But I mean, the normal thing we talk about then is just containing it in some way. So we use something like this for our organisms. You have a containment vessel. We have some way of containing it within a building or whatever else. The problem is, when that fails, it's like the gloves failing. It's done, that's it, everything's free. So we want something that's better. We want something that does that. And what I mean by this is you make the organism completely dependent upon you. You can make it dependent upon a chemical, something synthetic that it won't find in the wild normally to be able to survive. You could integrate it into its reproductive pathways, or you could even give it a half-life so that it can't survive for more than, say, an hour outside of the lab environment. There's all sorts of things we can do like that. And by smartly thinking about what we're doing, we can ensure that we contain these things. So moving on to the next sort of safety issue is things like this. If there's any Americans in the audience, I know they may struggle with some of this. MRSA is an antibiotic-resistant bacterium. It's what's responsible for a lot of superbugs in hospitals. It's very difficult to treat. And this arises because of overuse of antibiotics, effectively. So every time you take a course of antibiotics, it kills off the bacteria, but it doesn't kill off all of them necessarily. Sometimes there'll be some that survive because they're resistant to the bacterials, well, to the antibiotic. And the problem is, when you're modifying bacteria, you need to use antibiotics. So how do we deal with that? Well, the normal way we do it, again, this would be the sort of first thing, is we use friendly bacteria, or non-pathogenic ones, things that are unlikely to be a problem if they do become resistant. But again, this isn't really solving the problem. So the next thing we could do is we step it up a bit. We go on to not using antibiotics. We try and work with the components of the bacterium that we need to test. So if we can get just the component parts of the cells themselves and put them in a test tube and test it that way, that'd be far better. But again, when you're talking synthetic biology, that isn't the best solution. So instead, we go with something that is always popular when trying to destroy anything, and that's killing it with fire. As much heat as possible, it's bound to get rid of it. The fact is, there's very few organisms that can survive a good 200 degrees Celsius. The few that do aren't harmful, they're actually quite interesting to study. They're quite cute, they're called tardigrades. They also survive in a vacuum. Do look them up at some point. But yeah, so we normally just burn it as much as possible. So I mean, in terms of health and safety, that seems a pretty good method. And I said the ethics sort of bends into this as well. So on the ethical side of things, we thought, well, we can sort of treat ethics in the same manner. So what about some of the ethical issues that come up? And one of them we thought is actually the economic impact. A lot of people don't always think about this when they're dealing with like new engineering, because we have a general idea that a new technology will bring about new wealth. It will increase the size of the economy. It will bring about new jobs, everything else. But we don't always know that. And a lot of companies just go straight into it and don't know what's going to happen. And an example of this actually is a current iGEM team is working on a project that produces synthetic palm oil from bacteria. Now, palm oil is a major cash crop in Malaysia. It's the backbone of their economy. And it's used a lot in industry. The thing is, they're developing a system where they could actually make any unique blend of palm oil you could possibly want for any application whatsoever. So it's very lucrative in that respect. It's a lot cheaper because you don't need fields and fields of palm trees. And you could do it anywhere because it uses bacteria in an incubator. So potentially they could destroy Malaysia's economy with this project. But the good thing is the team's actually thought about this. And what they thought was, well, to do a little plot like this, and thought, how do we impact things? And their solution was, well, if we do make this, we should take it to Malaysia, and that way we can just replace their economy with it, but employ the same people that would have lost their jobs in working in our factories now to produce the palm oil. But it's at least by bothering to think about these things, you start to actually be a little more ethical than most current businesses, to be fair. I mean, I don't know about anyone else, but I'm not keen on the banks at the moment. I don't think anyone else has been for a while, but we can't say they're that ethical a lot of the time. So at least by bothering to think about it, we might be getting there. So this seems to be working well. Let's move on to another one. What's the next one, Dan? Oh. Yeah. This gets thrown around a lot, doesn't it, in science? Playing God, or going against nature, or it's something that's unnatural, or there'll be unforeseen repercussions. Incidentally, I really hate that phrase, because if they were foreseen, then for them to happen, that's a really bad thing. That's not good science. But the truth is that science isn't good or evil, it's the people that make use of it who could be doing something that's good or evil. But the issue with something like this is, I don't know what that means when you say, I'm playing God. I mean, does it mean this? Does it mean making a cow? I mean, we've bred these for years. In fact, cows never used to be that big. Cows used to be about knee-high. They're very small things originally, it's just we bred them for meat and for dairy. So, arguably we've fiddled there on playing God. Or maybe it's this. Maybe it's building skyscrapers and so on. I mean, they didn't used to exist before humans. I mean, is that playing God? Oh, no, I know. How about this one? Is that playing God? Jumping from the edge of space, landing and not dying. That seems pretty godlike. I don't know. But the point is, the issue here isn't really, just someone having a problem with what I'm doing. It's the fact that I don't know what they mean. So the normal way we deal with this in science is we tend to ignore you. But what we should do is talk, communicate. I mean, if this is me over here, and this is someone that thinks I'm playing God, we've got some meeting ground. I mean, we're both human, and I don't believe they're evil, and I'm fairly certain they don't believe I'm actually evil. But they may not understand everything else that's going on in my head. And that's kind of where the problem comes in. So the solution we've come up with here is trying to do more stuff like this. This is where iGEM is really good, is more outreach, talking to people. Although admittedly, I'm afraid you're kind of the wrong crowd. The problem is the people we need to talk to are your children, or your children's children, because they're the ones that will have the most clout on this in years to come, I'm afraid. Such is time. I'm very sorry. We'll try and do something about that, I promise. But moving on. So that's sort of the ethics and health and safety thing that we've gone through. But I now want to talk about the future, and I'm not going to make wild guesstimations and things and suggest that in 20 years' time we'll all be eating bacterial soup or something. I might do later, but you can mock me for it in 20 years' time if you want. I'm going to talk about the immediate future, and the first thing is the problem that synthetic biology has, which is a public image issue. We've inherited it from genetic modification, basically. So as I said, this is just a general Google search of different articles and things, and you can see up here, life as we grow it, the promises and perils of synthetic biology. Will synthetic biology become a GM-style battleground? Synthetic biology, playing God, there it is, I told you it's there. It's always there. And finally, updates, news and releases says, synthetic biology, big promises backed by bad theory. That's a little offensive, it's not too bad, it's working mostly. But the problem is it doesn't come across well, does it, if you can Google stuff like that. On the upside, it's slowly improving. The BBC, for example, is doing a lot more science programming these days that actually gets to the hearts of issues and tries to explain things. But as I said before, the issue here is really more sort of education and talking and communication, being open and doing what people haven't done for years, which is actually talk. So it's slowly improving, and it might get there in a few years' time. And I'm hoping more events like this would help with getting us there sooner, because we do everything we can to avoid these sorts of problems. We don't want to bring them up. But the other big issue, actually, that may not have a solution at the moment, sadly, is intellectual property and patent law. So the Human Genome Project, several years ago, back in the 90s and things started up, and a lot of private enterprises got involved. So they put in all the investment to help sequence the human genome. Now this is useful for medicine because a lot of the genes they found had stuff to do with congenital heart disease, potentially finding ways, developing cancer treatments or detecting cancer, and all sorts of things like this. The problem is, to make their money back, they decided to patent those genes. So you can have a congenital heart problem, of which a company owns the patent for the genetic code for that. you have the genetic code, but you don't actually own it. That company does. So that's why the Supreme Court in the US recently ruled that you can't do that anymore. In fact, all those previous patents are null and void. You cannot patent now any naturally occurring gene in the environment. Synthetic ones, different matter, but they've been designed. But any naturally occurring ones you can't do. So the problem is all these companies now that said we need to patent these to get our money back and carry on funding this research can't do that anymore. Which then begs the question of, well, what do you do? I mean, we're not going to innovate anymore. I mean, this will shut down, won't it? But instead what's happening is that they're moving towards patenting the process they use to actually develop these things. Instead they're moving towards going, oh, we did this in this way that's quite unique, so instead we'll patent that. So that seems where we're going towards, but we're still not sure, because until things like the music industry sort itself out as well, we don't know what's happening with intellectual property, which is a little worrying. I'd certainly encourage anyone who wants to read up on it. But moving on, I'll now actually talk about the actual immediate future. And this is where I might get a bit pie in the sky, but I'll justify it, I promise. So the first one I'm going to talk about is up here. This glowing plant thing. This is from a website called Kickstarter. Is anyone familiar with that? Probably not, don't worry. It's, okay, a few people are. It's fine, not everyone knows. Kickstarter is a website that crowdfunds things. So you literally put up a project, you explain what you want to do, you put out some incentives, and you basically seek investors through the internet. But they can invest as little as they want or as much as they want, and there'll be different amounts that you set and say, oh, for £5 I'll do this for you, for £10 I'll put your name on it. In fact, these guys for 50 quid were offering you some of the actual seeds for the glowing plant. Now their actual project, and this is quite cool, is they want to develop plants that glowed so profusely that you could replace street lamps with them, so they didn't need electricity and actually produced oxygen. Now the funny thing is, if you look up here, it says $484,013 pledge of their $65,000 goal. So to do this, they only needed $65,000. They've nearly reached $500,000. This is happening. This isn't just, it's not, pipe dream or whatever else. No, this is happening. This is currently happening. They've set some of the seeds out and everything. So that's one thing that's happening. Another thing I want to talk about are biohackers and sort of DIY bio. So this is for the London biohack space. I'll try and stop covering with my shadow. But the London biohack space, this is where amateurs, and as Dan said before, people that are more like the PC homebrew market, get involved. And they'll get into it and go, okay, I want to understand how this works, so I'll hack it, I'll take it apart, I'll try and figure out how it works. But they're now doing this with biology, because a lot of things that you need for it, like microscopes and so on, you can actually make yourself quite effectively and so on. There's even one in Leeds that's starting up soon. In fact, next Wednesday there's a meeting going on for it down on Regent Street. So do look that up. It's called Assembly, A-S-M-B-L-Y. But yeah, these groups are getting involved as well, because it's getting easier with things like iGEM, and iGEM as well as bringing in a DIY bio section. It's actually getting easier to do that as well. So there's all sorts of groups around the world that do this. There's one in Manchester as well. But the final one I'm going to mention is, of course, what current iGEM teams are doing. Now the thing is, iGEM teams often look at the big world picture. So they want to solve big world problems. They want to solve poverty. They want to deal with sanitation like us. Or they want to develop a t-shirt that is not only self-cleaning, but completely stain-resistant because it's covered in bacteria that will eat anything that would stain it. Things like that. But one of the coolest ones, this is from actually about 3 years ago, this shot. They're still in the competition, they've been carrying on with this, they're now sponsored by NASA, because what they wanted to do was develop bacteria that could terraform Mars. They want to send bacteria into space, further than any man has ever gone before, land on Mars, and turn it into Earth. It is actually possible, because it's basically what happened on Earth, is bacterial life and other organisms developed and changed the ecosystem. But yes, they're serious about this. They've actually gotten quite far along with it, and as I said, even NASA are sponsoring them. So what I'm really trying to say with this is, although some of it may seem quite crazy and far off, these are happening. This is actually happening now. So when we say it could be £72 billion for this sort of thing, yeah, it's likely. So all I want to do is, just as we move on, is I want to leave you with one little thing, which is, when you think synthetic biology, first off, think micro, and then think macro. Think first thing.